Just about ready to start, so why don't you all find a seat? Okay, it gives me great pleasure to invite you to the second week of the Oceans Alive seminar series. And tonight is the science showcase for local science fair winners. And we have 6 very talented high school students who will share their results of their science projects with you this evening. We'll have 3 speakers, and then we'll also have poster sessions out in the lobby following the talks, in addition to a demonstration here in the front. So there'll be lots of opportunity to meet the students, ask them questions, and share their ideas. On your way in, there was an orange sheet of paper called a Scavenger Hunt. And you're all welcome to answer the questions to the Scavenger Hunt. There's, on one side, is for grades 5 and up. And on the Junior Scavenger Hunt is for kindergarten to grade 4. You're all welcome to answer the questions. If you feel you can't tackle, even adults can't tackle the higher one, you can do the junior one if you like. But only the children will receive a prize at the end of the evening. So please, all of you, answer the questions, and the children will get a prize when they finish the scavenger hunt. So this evening we are here to honor Nate Twitchell and Timothy Cronin of Falmouth Academy, Adam Rago and Brian Gallagher of Falmouth High School, and Carolyn Trask and Stephen Baumfock of Mashpee High School. Our first speaker this evening will be Adam Rago. Adam is a sophomore. He will present his project, Interactive Effects of UV and Natural Pesticides on Mealworms and Crickets. And for his efforts, he won the $2,000 Mary Sears Scholarship from the Woods Hole Oceanographic Institution, and he will be participating in the International Science and Technology Fair this weekend in San Jose, California. Adam?
Hi, as stated earlier, my name is Adam Rago. I'm a sophomore at Falmouth High School, and the title of my project is Interactive Effects of UV and Natural Pesticides on Mealworms and Crickets. Since the introduction of DDT in the 70s, which had a very high toxicity to the animals in the environment it's sprayed, applied to, and low selectivity as to what's killed, it has led to the advent of natural pesticides, that is, insecticides that are derived from natural sources, namely plants. Depending on how it's applied, light can have a different effect on the pesticide, making it either more toxic or less toxic. If sunlight is applied to the chemical, it can photodegrade under natural conditions. But if the animal is to eat the pesticide and then is exposed to UV light, the toxicity can become much more great. In my experiment, I wanted to test the effects of the natural pesticides through a series of experiments. The first one was a direct contact experiment to test the acute toxicity of the pesticides in concentration from full strength to 1 part in 100,000. The next experiments were to test the interactive effects between UV light and the pesticide through a series of ingestion and interactive experiments. The third set of experiments were deactivation experiments to see if the pesticide could be photodegraded to a sublethal state. To test the interactive effects in this experiment, I used a UVB light shown here. That it was applied at 0.5 to 3.5 microwatts per centimeter squared, which is about 3 times the UV content in the summer. On the ultraviolet spectrum, the UVB light is from 290 to 320 nanometers, which is the teal box on the graph. The animals used in this experiment, mealworms and crickets. The mealworm is Tenebrio molitor L, and the house cricket, Acheta domestica, was also used. Both of these animals were used because they're easy to maintain, robust, also inexpensive, and they eat on a frequent scale. The natural pesticides used in the experiment are 4 of them, 2 synthetic ones that were purchased at a garden store, and then 2 plant sources. Neem and pyrethrin were purchased at Mahoney's Garden Center, and marigold and St. John's wort plants were used alone. All of these pesticides have a low toxicity to humans. The LD50 value, that is lethal dose where 50% of the test population is killed, for all of them is relatively small. Azadricin, the active ingredient in neem, has an LD50 of 5,000 milligrams per kilogram. For pyrethrin, it is 2,000 milligrams per kilogram. Both of those tested on rats. Alpha-trethionyl, the lead ingredient in marigolds, has a toxicity of 800 parts per billion, tested for mosquitoes. And hypericin, the active ingredient in St. John's wort plant, has a toxicity of 16 micrograms per gram for the Manduca sexta animal, which is a tobacco hornworm. The first experiment I set up was to test the toxicity of the chemicals in concentration and with full strength. Neem and pyrethrin chemicals were used in this test, and they were applied in 10-fold dilutions from full strength to 1 part in 100,000. They were applied with a direct spray of 1 milliliter to the test population of 20 mealworms. The results of this test are shown in these graphs here. Each line represents a different concentration of the chemical, shown in the legend on the right of each graph. From this experiment, I concluded that natural pesticides are effective even at very low concentrations. In these tests, all of the animals were killed at the end of a 48-hour observation period, where they were observed at intervals of 5 minutes, 1 hour, 24 hours, and 48 hours. I also concluded from this experiment that as concentration decreases, the lethal effects also decrease. That is, the full and high concentrations of the pesticide killed the animals mostly within 5 minutes or 1 hour, while the higher concentrations of the lower concentrations of the pesticide took a longer time to kill all the animals. The next experiment set up was an experiment to test the interactive effects of the UV. The next experiment was set up to test the effects of neem and pyrethrin by application to the food of the animals. The chemicals were soaked, a piece of lettuce that the mealworms fed on were soaked in neem or pyrethrin, again in the 10-fold dilutions from full strength to 1 in 100,000. There are 20 mealworms used in each test. And the results of this experiment are plotted in these graphs, with neem on the left and pyrethrin on the right. From ingestion experiments, I concluded that direct spray is more deadly than food source application. In the ingestion experiments here, not all of the animals were killed in most test groups, while all of the animals were killed in direct spray tests. I also concluded from this experiment that the results suggest that chemicals have an anti-feedant effect, meaning they discourage animals from feeding on the plant that they're applied to. This anti-feedant effect could also be due to the ability for animals to avoid contact or to reduce feeding on the chemical. So there's results that suggest an anti-feedant effect, but it can't truly be concluded to. The next experiment set up was to test the effects of the chemical on the house cricket, Acheta domestica. The test chemicals were neem and pyrethrin, applied at 1 in 1,000 and 1 in 100,000 concentration. These toxicities were determined for use on crickets based on the mealworm experiments conducted before. Again, they were applied with a direct spray of 1 milliliter of the respective solution to each animal in the test. The results of these experiments are shown here with, again, neem on the left and pyrethrin on the right. From these experiments, I concluded that natural pesticides are less toxic to crickets than they are to mealworms. Not all of the crickets died as a result of direct spray tests, while more crickets died, or more mealworms died, in the same direct spray test. The next part of the experiment moved into the effects of interactive between the pesticide and the UV light. For an ingestion experiment, all 4 natural pesticides were used: neem and pyrethrin that were mixed with the animal's food source at 1 in 1,000 concentration, and marigolds and St. John's wort, which were fed to the animals in plant form. The experiment was conducted over 5 days, with the first day being a starvation period, the second day being a feeding period, and the third day being UV exposure. After the animals had ingested the chemicals, they were exposed to UVB light for 24 hours at 0.5 microwatts per centimeter squared. The results of these tests are shown here for neem and pyrethrin, and here for marigolds and St. John's wort. From these experiments, I concluded that the effects of the chemicals were significantly more deadly when animals were exposed to UV light after ingesting the chemicals. For example, in all these experiments, a control group of ultraviolet light only, no treatment at all, and chemical treatment only did not kill any of the animals, which are the 3 graphs shown at the top of the graph. But when the chemical interacted with the UV light once inside the animal, many more animals were killed. In the neem group, more than half of them were killed, as shown in the red bar. The same is true for marigold and St. John's wort treatment. The red series drops off farther than the control treatments alone. Unfortunately, the same effect was not shown in pyrethrin experiments. The phototoxic effect was not able to be shown. The next and final experiment was a deactivation experiment to see if the chemicals could be photodegraded under UV light, which is, according to reading, is said to have happened. Chemicals were exposed to UVB light at 3.5 microwatts per centimeter squared for periods of 12 and 24 hours to all of the chemicals before the test. Then they were applied to the animals, and the effects were noted. From these 4 experiments on neem on mealworms, pyrethrin on mealworms, neem on crickets, and pyrethrin on crickets, it was concluded that the deactivation was partially successful. That is, the chemicals deactivated longer, killed the animals at a slower rate. All the animals ended up dying for mealworms at the end, but the rate at which they were killed was slower. For example, in the upper left-hand corner under the Neem test, the blue is the full concentration that was not exposed to ultraviolet light at all, which killed most of the animals within 5 minutes. Rather, the yellow or orange series is pesticide that was deactivated for a 24-hour period. The time it took to kill all the animals was slower, but in the end, all the animals still died. To further apply my studies, possibly next year in a phase 2 experiment, I'd like to lengthen the studies to accurately assess the long-term effects, that is, longer than a 48-hour period. I'd like to dilute the chemicals more to find a direct spray solution that would have a sublethal effect on the animals. I'd like to make a larger group of test subjects for results that are more accurate. If there's a larger test group of animals, there would be a more accurate result. I'd like to focus on the interactions between UVB light and the natural pesticides, that is the interaction between light that makes the chemical either more or less toxic. And finally, I'd like to examine the effects of sublethal concentrations of the chemical on the growth rate, reproduction, and development of animals, which would be a longer-term test of the effects of the chemicals. I'd just like to thank my dad, Dr. Paul Rago, for a lot of valuable help on the experiment, and Dr. Steve Murawski for loaning me the UV light used for the experiment. Are there any questions?
We'll be trying to interact with Adam at the end of the talks as well, but if anyone has a question, ask now.
Go ahead.
Why did you pick that one?
It started as an experiment that was going. I dealt with the mealworms last year in a different experiment, and I was using the same animal in this experiment. As the mealworm grows, it sheds its skin. So I was originally going to look at a pesticide that stops the animal from growing or shedding its skin. It turned out to be a controlled substance, so I kind of stumbled upon the natural pesticides. Yes?
I'm unclear as to how you diluted these commercially purchased materials.
They were diluted with 1 milliliter of the chemical was mixed with 10 milliliters of purified water, and then there was diluted serially in tenfold dilution. So 1 milliliter of the stock solution was put in 10 milliliters of water to make a 1 in 10 concentration. Then to make a 1 in 100 concentration, that was 1 milliliter of that was put into 10 milliliters of water.
The primary material was a liquid, is that correct?
Right.
It wasn't a solid.
No, they were not diluted. They were liquid dilutions.
Did any of your experimental subjects escape?
Actually, in one test, some crickets, they were held in a petri dish with saran wrap over the top, and they actually chewed through that and escaped. So I had to put them in plastic bags for future tests, but that only happened in one test. Yes?
Carry the experiments at your home, most of them?
Yes, they were all done at my home in my basement.
Thank you, Adam. And Adam will be available. Our next speaker this evening is Nate Twitchell, who won the top honors at the Falmouth Academy Science Fair for his investigation of the genetics of wild oyster populations. He compared 2 oyster populations from Martha's Vineyard with 2 from Falmouth. For his work, he was awarded the A. Lawrence Pearson Scholarship from the Woods Hole Oceanographic Institution, and he will also be traveling to the International Science and Engineering Fair in San Jose, California this weekend. Nate?
Before I get started, I would just like to thank Dr. Alan Kazarian as well as Dr. Hemage Carmen for allowing me to use their lab space down at MBL right here in Woods Hole. Without their help, I wouldn't have been able to carry out this experiment. And I would also like to thank my science teacher, Mrs. Victoria Centrino, for helping me look into the statistics as well as as reading many drafts of my paper for my project. The title of my project was Population Genetics of Local Wild Oyster Populations Using PCR. Now, the reason I actually got the idea for this project is I've worked on an oyster farm since I was about 12 years old down on Cuddyunk Island, down the coast a little ways. I've always been very interested in oysters. Actually, last year I did a project where I took 2 cultivated populations of oysters, which we have down on Cuddyunk, and I compared them. And what I actually found was that these cultivated populations, one from Maine and one from New York, were almost exactly identical. And I was quite interested by this, so what I actually thought would be interesting for this year is to take several wild populations locally and see if they were as similar as these populations or if they showed more differences. The populations which I took were from one population was from West Falmouth Harbor, which is right here. One was from Quisset Harbor down the road a little ways. And the other 2 were from Tidsbury Great Pond, one on the Chilmark side and one on the West Tidsbury side. Now, the purpose of my project this year was, let's see if I can keep from losing all this. The purpose of my project this year was to determine how either heterogeneous or homogeneous these populations were by using genetic fingerprinting to see any differences between these populations. Now, what I hypothesized was that overall these populations would show to be fairly heterogeneous, but that there would be some noticeable similarities between populations from the same town, meaning that the 2 Falmouth populations would be fairly similar to each other, and separately the 2 Vineyard populations would be fairly similar to each other. Now, the process which I used to actually carry out this project was called rapid PCR, and what that stands for is random amplification of polymorphic DNA using polymerase chain reactions. That's a real mouthful, but what it actually is you first do a DNA extraction. I used chloroform, a double chloroform extraction to actually get out the DNA. Then I precipitated it out using isopropanol and purified it with ethanol, and then diluted it with tea, which is a tryst buffer. And then I ran a set of quality gels, and what these quality gels allowed me to see was if the DNA was still usable or not. So then I was able, through finding out the usable DNA or if the DNA was degraded, to pick the oysters that I actually wanted to use for my final PCR study. Now, in my PCR study, I picked 2 primers that I wanted to look at my DNA with. I picked primer 101 and primer 103. Primer 101 was really simple to pick because I used it last year in my project, and it showed a really nice banding pattern, so I thought and figured that it would show a good one again this year. And I picked 103 because Dr. Kazarian, who I was working with, had just finished a study with bay scallops, and it showed really nice banding patterns. So he and I both thought that that would be comparable to oysters. So what I actually came out with was gels that looked like this. And this is two of the gels that I had from primer 103. And if you can see here, each one of these lanes right here is a different organism, and each one of those bands right there is a different base pair. Now, once I had all my gels, I actually needed to get my data off them. So what I did was, this won't be easy to see, but it will give you an idea of what I did. I took all the pictures that I had taken of my gels and I taped them together in a fashion like this. And in each of these gels, as you can see down here, I had a marker. And what the marker allowed me to do was have a 0 point for my data. And what the 0 point meant is that I could take all the data and hang it off a 0 point and then do my band counts off of that. And what I did for my band counts is I picked the first band that I could clearly see, and I designated that band 1, and then I counted down from there throughout all the bands that I could see. Once I had all my bands taken and counted up, I actually made up charts. And what this chart shows you is the band and then the frequency, or the number out of 100 that would actually have, the number of oysters out of 100 that would actually have that band. And now, once I had all these frequencies, I wanted to be able to determine how similar or different these populations might be to each other. So I did a series of t-tests, and what I found was, first I did a set of t-tests within the Falmouth populations. I compared both Falmouth populations to each other. I ran a t-test for both primer 101 and 103, and I found that there was no significant statistical difference at the 0.5% level of uncertainty of my result between the Falmouth populations. So then once I determined that, I went and I compared the oysters from Tidsbury Great Pond, and what I found as well, running a t-test for both primer 101 and 103, was that there was no statistically significant difference at the 0.5% level of uncertainty of my result between the Tidsbury oysters either. And what this allowed me to do then was compare the populations from the vineyard to the populations from Falmouth. And what I found then was that there was, between Falmouth and the vineyard, there was a statistically significant difference at the 0.5% level of uncertainty of my result. And I thought this was kind of interesting, so what I actually did after I had finished this was I went back and I did some background research. And one idea that I have for the fact that the populations from the same town were so close to each other is when oysters are first born, they're in a larval stage, and they're free-floating in the water. So this is a 2-week period of time in which they're free-floating in the water, and they're at the will of wherever the currents may take them. And I think the reason that the two Falmouth populations and then the two Tidsbury populations were so close to each other is this is only about a 6-kilometer distance apart, and there's a strong current that runs along down Buzzards Bay all along here, so that would draw the larvae right out of West Falmouth Harbor and push them down to Quisset Harbor, and then conversely, on a flooding tide, it would bring the larvae right out of Quisset Harbor and up to West Falmouth Harbor. So I think this may be why any differences were canceled out between those two populations. And if you look at the Chilmark population and the West Tidsbury population, that's only about a 2-kilometer difference over an open harbor, and the oyster larvae could easily disperse over that distance during a 2-week period. So that may be why there are so many similarities between the populations from the same town. Now, an idea that I had for the reason that the vineyard may be different from Falmouth is that there are things called bridge populations. So what a bridge population is, if you're comparing a population from here and a population from here, there may be 2 or 3 or numerous populations in between. And what that means is that if this distance between the 2 populations that you're comparing is too great for the larvae to actually travel during that 2-week period, they may be able to travel somewhere in the middle and settle out and then breed there and then get dispersed from there the next year on to the next population, which if there were bridge populations, that would cancel out any differences. But due to the fact that Vineyard Sound is quite deep and there are quite strong currents, oysters like to grow in about 5 to 10 feet of water at the deepest and in tidal zones. So Vineyard Sound is not very conducive for oyster growth, and therefore there aren't many bridge populations in that area. Now, what I feel I can conclude from this project is that populations from within the same town showed no statistically significant difference, and they're very similar. But when you go on a more regional scale, there are more noticeable differences. But I think it would be interesting to expand upon this project and take populations from up and down the East Coast and see if this trend that within a localized area the populations are fairly homogeneous, but as you spread out and deviate from that area they become more heterogeneous, and see if this is a natural trend or if this is just a localized situation due to the tides and currents and geographic situation that we have right here in Falmouth. Can I entertain any questions? Yes?
Why do you like it so much?
I think the reason that I like this project so much is that I've just been working with oysters for so long, and I work with them all summer long, and it's a fun job. It's a pretty relaxed job, and it was fun to continue it into science. Yes?
How long does the oyster larva stay up in the pond?
How long does it stay up in the floating stage?
Yeah.
About 2 weeks. That's what I read in the book. Can I entertain any other questions?
Nate will also have his poster out in the lobby for discussion afterwards. Thanks, Nate. Our last speaker this evening is Stephen Baumfak, who is a senior at Mashpee High School. And he won 3rd place at the high school fair for his project, The Effect of Temperature on the Rate of Digestion of the Protein Albumin. And Stephen will be attending Liberty University this fall. Stephen?
I use the protein albumin, which is from eggs, and the enzyme pepsin, which you can find in your stomach, and hydrochloric acid mixed with distilled water to digest the protein albumin. Is that on? Anybody? Okay, so in essence I created a stomach in a jar with hydrochloric acid, an enzyme, and a protein. And what I wanted to do was vary the temperature to see if a lower temperature or a higher temperature would change the rate of digestion. Right here, this is a graph of the rate of reaction of the enzyme pepsin. And according to this graph, at 10 degrees Celsius, the rate of digestion or the rate of activity of pepsin is very low, and the same at 60 degrees Celsius, and then around 43 is your maximum rate of reaction. And I use 37 degrees Celsius, which is human body temperature, for my first temperature, and then I varied down to 10 and up to 60 because of what the graph told me, and I just wanted to see if that was right. And the reason why I did this project was because we did an experiment with pepsin and albumin and hydrochloric acid in class, and the test didn't go well, and we couldn't see the colors right, and I just wanted to dig in a little deeper. I also used a solution called biuret reagent, which is a blue color in the presence of enzymes and proteins, and then when the enzymes and proteins get broken down, it changes color to pink in the presence of polypeptides and peptides, and then it changes even more color if it gets broken down all the way. So what I did for my experiment was I put all the solutions in the jar, and I used a water bath to heat it up to 37 from room temperature, or down to 10 I used ice. And well, what I did was every half hour, because it takes a while, I took a reading on the spectrophotometer, and I used a wavelength of 420 because that is roughly purple, which is the color biuret reagent is in the presence of proteins. And so what I was trying to show was that the color was changing. And here are my graphs of the different temperatures of absorbency and transmittance of the light, which is what the spectrophotometer measures. I also, if you can see here, I took 3 trials at each temperature and then made an average, and that's what's on the graph. So, wow, I haven't presented this in a while, so I'm kind of losing myself. At 37 degrees, which is this line here, and this line coming down on transmittance, what happened was the temperature changed, or not the temperature, sorry. The color changed, and the spectrophotometer measured the change in wavelength here. And it's the absorbency. It measured the absorbency of light through the solution, and that shows the color was changing. And then at 10 degrees, I had a flat line for both, which shows that the color wasn't changing, and that means that the proteins weren't being digested. And then I bumped it up to 60, and I got a little bit of weird results in the absorbency and transmittance, which I'll explain later, but it went down to negative numbers in both. So what I showed here was at 37 degrees Celsius, which is human body temperature, and it's very near the top of this bell curve, which shows it it's supposed to be a high rate of activity. The absorbency increased, transmittance decreased, which is they're just inverses. I just measured both to make sure everything was all set. And that just showed that the color was changing, so the light wasn't going through. Or the transmittance went down, so yeah, the light wasn't going through. It was being absorbed. And that showed that at 37 degrees it digested, which is what I predicted, because if you eat an egg, you digest it, because that's what your body temperature is. It just kind of makes sense. At 10 degrees, which is pretty cold, especially if you were a human, so it didn't digest. And one of the things that I was thinking about was 10 degrees Celsius could possibly be like the temperature, body temperature of a fish or something in the ocean. And obviously, yeah, they get to have protein somehow. And I kind of came to the conclusion about that they must have a different enzyme for digesting protein, because if it's at 10 degrees and they're using pepsin to digest the protein, it's not going to happen. And now we go to the 60 degrees. And since I started at room temperature and increased to 60, it had to climb the bell curve. So at one point it was reacting high, and then it came back down to low activity. And that's shown here by this slant line, which means that there was some digestion occurring, and then it cut off. And the interesting thing was that at 60 degrees, a precipitate formed. And I believe that the precipitate was the protein denaturing, which was the albumin, and the enzyme pepsin was just inactive. And the reason for the absorbency being off the charts is because the precipitate was in the spectrophotometer, and the light couldn't pass through it because it's not a liquid. There's like a little bit of solid in there. And so to prove or to find out whether it was the protein or the enzyme that was denatured, I could lower the temperature back to about 40, and if the pepsin wasn't denatured, then I could add more protein, and it would digest that protein and then change color because of the Bayer reagent reaction. And I think that's just about it. I got questions here?
Are you going to do this when you grow up?
Well, I don't know, actually. I wasn't very interested in science, and I didn't want to be in this biology class at the beginning of the year, and now it's my favorite class, thanks to Miss Abchez back there. Anybody else?
Why did you use this
Because we did something on it in class, and it didn't come out right, and so I wanted to fix it and make it a little better and see what happened. You, sir?
Did you use agitation to promote mixing?
Yes, thank you. I put it in the water bath, and then I had a little magnetic stir, put it on the hot plate on stir, and it stirred it up, pipetted it out, and put it in the cuvette. Thanks for clarifying that for everybody else. Anything else? All right.
Thanks, Stephen.
I'd like to introduce our other three guests this evening. Brian Gallagher, if you'd stand up, from Falmouth High School. And Brian received a first place award at Falmouth High School for his project and entitled Fish Heat, Enthalpy Changes During Aerobic and Anaerobic Metabolism in the Killifish. And Brian will be explaining that project in the lobby after we break here. Carolyn Trask, would you stand up? Carolyn is a freshman at Mashpee High School, and she received the top scholarship from the Woods Hole Oceanographic Institution, awarded for her project, a study of the caloric content in a variety of commercially prepared brands of Acus hypogea, which is the peanut. And Carolyn will be presenting her project. But this contraption that you see up here will have a live demonstration by Tim Cronin, who doesn't want anyone to leave the auditorium until he's shown his project. And Tim is a junior at Falmouth Academy. His project was on improving the aerodynamics of pickup trucks. And for Tim's project, he received an award from the Marine Biological Laboratory and a scholarship from the Sea Education Association. And Tim, would you like to make a presentation or demonstration of your project?
Okay, well, what you see here is a wind tunnel that I built to test the aerodynamics of pickup trucks. Basically, I started out with the idea of building models onto the back of the trucks to make them more streamlined. This one here was used as a control. I also tried with the tailgate off. I have a model in here right now. I'll show you. This is simulating the tarp that is available to put over the back of a pickup truck. This here is just linear straight backed. And this design, as you can see, this one, and then this last one, which most closely simulates the design of a normal car. I'll turn this on and I'll show you how it works. Basically what I have here is a force sensor, which measures the amount of force applied in this direction by the pickup truck. This is a leaf blower that's turned on to the mode of suction. And I have a transformer here which can vary the voltage. Right here I have an airspeed indicator which was used to determine the air velocity at different transformer settings. And then transformer settings were used in all the graphs and analysis of data. Let's see what else. This, I have the graphing calculator here, and underneath is a data mate, which converts the analog information which this sensor gives to digital form that I could read out and record as data. Turn it off. No, unfortunately, if you could come closer, you'd be able to see the pickup truck right here. It's blocked by this support here for the wind tunnel. If anyone would like to come down afterwards, you could see this. Basically, what I found in this experiment was that these two models, the linear-backed and the sedan-like, appeared to best reduce the drag coefficients. This model was the worst, and which is what I expected it to be. I did a statistical analysis and found that at higher speeds, especially, there was a much larger difference, especially for these two models. And it was concluded that this was the best model to put on the back, as it could increase average gas mileage by around 3 miles per gallon. With a pickup truck that gets about 20 miles per gallon on average. With gas at approaching high prices, it becomes much more economical to conserve gas that way, and of course, it's also good for the environment. If you had a truck that was put 100,000 miles on it, you could probably save around $1,000 over the life of the truck on gas alone if you use this model. Which is why I think this is relevant. Future studies for me could include inducing smoke into the wind tunnel to observe the vortices and eddies formed in the wake, and observe the differences between the different models and how the air flowed over them. And also building real-life size models to put on the back of a real pickup truck and test whether it actually did increase the gas mileage. Any questions on this? Yeah? Well, I chose this project because my dad posed a question to me. He was wondering what would be the effect if you put something on the back or just drop the tailgate off or anything like that. And so I thought it would be interesting to investigate it, and I did this on this scale to be able to get a lot of data and test it. Yeah.
I'm curious. If I take the tailgate off of my pickup truck, what am I going to see for improvement in mileage, gas mileage?
I didn't notice much of a difference at low speeds, and only at the very highest speeds did I notice any significant difference at all. I'm guessing that at highway speeds, you might be able to get about 1 mile per gallon extra, if that. If I wanted to do that, I'd probably use a bigger wind tunnel and more precise trucks, because these are really rough. As you can see, the bottom of them are not realistic at all. Yeah?
How long did it take you to do this?
Well, the data collection took me a while. I have pages and pages of data in here, but the building process went relatively quickly, and on the whole, it probably took me weekends in January and February to do this. Yes?
Did you ever think of reversing it and putting the thing on below and having it connect the other way so you could test the aerodynamics of the front of the truck?
this is testing the aerodynamics of the front because it's pulling it through. The air is being pulled through this way. But basically, why I chose for suction was that there was a higher cubic feet per minute rating, meaning that I could get this up to its top speed, which was about 65 miles an hour, and also because when you are on suction mode, it creates less problems. There's much less turbulence because it's being pulled through much better. Yes?
Roof racks also, it's been proven will cut your gas mileage way down. So not way down, but it does make significant difference.
I might want to test it on roof racks too.
Well, I mean, they know that's
Yeah.
a fact, right?
Yes?
Do you think there'd be any difference if you widened the tube?
I'm sure I'd see a difference if I probably wouldn't see too much of a difference with this, but I bet I'd see a difference. The actual drag coefficients and the comparison would the actual drag coefficients would more nearly approach real values if you used a bigger tube and a much more realistic truck. I had looked at there were some very nice model pickup trucks, but they were too big to fit in this tube, and I couldn't find a fan with a high enough cubic feet per minute rating to get it up to highway speeds. Yes?
If you increase the size of the tube, you might very well want to try to position your truck in the center of that tube to get away from any side effects that are
Yeah.
on tubes.
I had contemplated putting it on a pedestal. I thought that putting it on the base of the tunnel would more nearly simulate the ground effects during normal driving, though. That was something I considered. Any other questions? Thank you very much.
Well, Tim and Stephen will have their projects up here. Carolyn, Nate, Brian, and Adam will have their projects in the lobby. So please take some time to ask them questions so you can fill out your questionnaire. Thank you all for coming this evening. The last lecture in our series for the spring will be next Tuesday night, and will be presented by Dale Levitt and Brad Morse on There's Something Fishy About That Cranberry Bog project looking at conversion of retired cranberry bogs for fish culture. So the scavenger goes to Tracy or Sherry. And again, if